In this video, I'm comparing the differences between eukaryotic and prokaryotic transcription. Where the transcription takes place in the cell. In eukaryotes, as the DNA is present inside the nucleus by a membrane-bounded nuclear membrane, the transcription in eukaryotes takes place inside the nucleus, while in prokaryotes, the DNA is present in the cytoplasm and there is no membrane bounded by this DNA. There is no nuclear membrane present. In prokaryotes, the transcription takes place in the cytoplasm. RNA polymerase. In eukaryotes, there are three different types of RNA polymerases for the synthesis of three different types of RNAs. These are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 synthesizes the ribosomal RNA. RNA polymerase 2 catalyzes the synthesis of messenger RNA and snRNA, while the RNA polymerase 3 it catalyzes the transfer RNA and 5S subunit of the ribosomal RNA. In eukaryotic transcription, these three different types of RNA polymerases are required for the synthesis of different types of RNA. While in prokaryotes, only a single RNA polymerase is required for the synthesis of all these different types of RNAs. As you can see here, in prokaryotes, the RNA polymerase consists of 5 subunits. These 5 subunits are 2 alpha subunit that is responsible for binding these RNA polymerases, beta subunit which is responsible for the receiving of new incoming ribonucleosides, and the beta prime it is responsible for binding to the DNA double helix, and the sigma factor it is only present in the initiation step. Once the initiation has been started, this sigma factor will be released. So this RNA polymerase for the prokaryotic transcription consists of 5 subunits, while the eukaryotic transcription these polymerases consist of 10 to 11 subunits. Transcription factors in eukaryotes. There are a combination of different transcription factors that form the initiation complex in eukaryotes. These transcription factors are transcription factor 2D, B, F, A, and E, and H. These transcription factors combine together to form a complex with the RNA polymerases. If these transcription factors are not present, they may affect the synthesis of RNA. While in case of prokaryotes, these transcription factors are not required. Promoter sequences. These are specific sequences that are present upstream of the coding region in both organisms, in eukaryotic as well as prokaryotic. These are specific sequences that are recognized by the RNA polymerases from where the transcription has to begin. These promoter sequences are different in eukaryotes and prokaryotes. In eukaryotes, these are cat box, which is present -80 base pair upstream, GC box, which is present -100 base pair upstream, Tata box, which is present -25 base pair upstream. This minus is for the upstream and plus sign is for the downstream. In prokaryotes, these sequences are present -10 base pair upstream and -35 base pair upstream. So these are the specific sequences to which the RNA polymerase will recognize and bind to them, and from there they will initiate the transcription process. The first process for the transcription is the pre-initiation complex. In eukaryotes, as the DNA is bind with the histone proteins. In eukaryotes, the DNA is present in the nucleus and the DNA is present in a condensed form. They are bound with the histone proteins and they are also present in the form of a nucleosomes. So these histone proteins has to be removed for the transcription process. So the transcription initiation in eukaryotes is a complex process. For that purpose, different transcription factors and together with the RNA polymerase and some activators are required. So there is the formation of the pre-initiation complex in eukaryotic transcription because of the presence of the histone proteins, while in case of prokaryotes, as the DNA is present freely in a loose chromatin form in the cytoplasm, there is no requirement for the complex initiation process. So there is no formation of pre-initiation complex in the prokaryotic transcription because the DNA of the eukaryotes is more complex than that of the prokaryotes. Therefore, the pre-initiation complex will be formed in eukaryotic transcription. Initiation in both prokaryotes and eukaryotes, there is the unwinding of the DNA double helices, and as the RNA polymerase unwinds the approximately 25 base pairs, and now these base pairs will be exposed for complementary base pairing. The only difference is that in eukaryotes, as it is a complex process, there is histone proteins. For the removal of histone, they require the presence of ATP. So in eukaryotes transcription, the initiation process is energy dependent process. They require the ATP. While in prokaryotes, as it is not a very complex, it does not have the histones, proteins bind to them, so it does not require the presence of energy, does not require the ATP. The next step is the elongation step. In elongation step, in both prokaryotes and eukaryotes, the nucleotides keep on adding, and there is the synthesis of the chain of the RNA. The RNA chain is synthesized as it is copied from the DNA, and it gets elongated until the termination region reached. The difference for the eukaryotes and the prokaryotes is that in eukaryotic transcription, when the RNA polymerase keep on elongating, starts the elongation steps, keep on adding the new nucleotides, the RNA polymerase, all the transcription factors will dissociate from the RNA polymerases to only except for the transcription factor 2D. As you can see here, transcription factor 2D, which has the binding factor transcription Tata box binding protein. This transcription factor 2D will keep on binding onto the promoter region, which contains the Tata region and the Tata box. It will keep on binding on this region, and the rest of the RNA polymerase will keep on synthesizing the transcription and RNA. While in prokaryotic transcription, the RNA polymerase will keep on elongating and move forward by unwinding the double helices and keep on adding, catalyzing the synthesis of the RNA, the sigma factor will be released. In prokaryotes, it will leave behind the sigma factor, and the rest of the RNA polymerase with the remaining subunits will keep on moving. While in eukaryotes, as you can see, RNA polymerase will dissociate all the transcription factors except for the transcription factor 2D. The final step is the termination step. In eukaryotes, when the termination occurs in eukaryotes when the RNA polymerases, while keep on reading the DNA strand, a specific sequences will come that will be known as the terminator sequences. These terminator sequences are lacked in the RNA polymerases, and then RNA polymerases will stop, will stall and dissociate from there and will release the RNA free. While in prokaryotes, when this termination region reached, there are two different methods that can be used. One is the row dependent termination. In row dependent termination, the protein known as row protein will be used. Row protein, it will slide the messenger RNA, and then it will reach to the region which are known as the termination sequences, and the termination region, it will cut the RNA and will release it free. In row independent termination, when the termination sequence is reached, this RNA will fold back and will form the complementary base pairing, and then by forming the complementary base pairing, it will lead to the formation of the hair loop formation. And this hair loop formation, the RNA polymerases will not be able to make the copy of these sequences and will dissociate from there. RNA polymerases will dissociate, and the RNA transcript will also be set free. And in these two methods, this row dependent method in which row protein is used, this is an ATP. It requires the presence of the ATP, while in row independent method, there is simple just hair loop formation because of these sequences are rich in uracil. When the RNA transcript is released along with the RNA polymerases, there is a difference in prokaryotes and eukaryotes. In prokaryotes, as the transcription takes place in the cytoplasm, so there may be no processing or there may be little processing. The RNA is synthesized in the cytoplasm, and then simultaneously at the same time the translation also occurs. So that's why no processing is required, or maybe little processing is required. In case of eukaryotes, the RNA is synthesized in the nucleus. It will be premature RNA, or it will be primary RNA. Then they have to go through processing and modifications. After that, they will be released into the cytoplasm. Before going into cytoplasm, the maturation steps include three things. There will be 5' capping, in which there is addition of 7-methylguanosine to the 5' end. There is 3' adenylation, as polyadenylate or polyadenine will be added. Many adenine nucleotide bases will be added to the 3' end, that is known as polyadenylate. And then when the RNA is synthesized, it will contain the coding sequences and non-coding sequences. The coding sequences are known as exons, while the non-coding sequences are known as introns. As you can see here, these intervening sequences are the introns. These are removed by the enzyme known as spliceosome. Spliceosome will cut out these intervening sequences, and then these exon, the coding sequences, will join together to form the fully coding transcript. Now this is the messenger RNA, which contains all the coding sequences for the formation of the proteins. After the synthesis of RNA, this RNA will be utilized for the synthesis of the proteins. How? As these RNA will be decoded by the ribosomes by adding the specific amino acids, and these amino acids will form the polypeptide chains, and polypeptide chains, after post-translation modifications, they will be responsible for the formation of specific proteins. In eukaryotes, this process is a discontinuous process in which the transcription takes place in the nucleus. In the nucleus, the primary messenger RNA is formed, which is then processed to form mature RNA, and then this mature RNA will be released into the cytoplasm. In the cytoplasm, the ribosomes machinery will recognize it and then will lead to the formation of proteins by the process known as translation. In eukaryotes, transcription occurs in nucleus while translation occurs in cytoplasm. In prokaryotes, it is a continuous process in which the transcription and translation both occurs in the cytoplasm. As the RNA formation takes place in the cytoplasm and the ribosomes machinery is also present in the cytoplasm, ribosomes will simultaneously come, and as the RNA is synthesizing, the ribosomes will also keep on translating these messenger RNA for the synthesis of the specific proteins. This was all about, I have compared the differences between prokaryotic and eukaryotic transcription. If you want to learn in detail about the prokaryotes and eukaryotes transcription, there are separate videos in which I have explained about the transcription in prokaryotes and transcription in eukaryotes.